Okay, good night. This video is going to look at some of the major topics in digestion, including the role of the teeth, the role of the various parts of the alimentary canal, the function of the enzymes that are active at various phases of digestion, as well as absorption, what that is, and assimilation, including the difference between digestion and excretion. Okay, so let's look just quickly for a few minutes, and then I'll also go over a few past paper questions on this topic. So when we look at the teeth, we just need to be aware, of course, of the various types, the molars, premolars, canines, incisors, and how the premolars and molars are the ones that assist mostly with crushing and grinding. The incisor and canine are cutting and biting off of pieces of food. The internal structure of the tooth is something that you should also know in terms of the outermost layer being the hardest layer, dentine, and pulp cavity, which includes the blood vessels and the nerves inside, and then the cement and the periodontal membrane that holds the tooth in the socket. Tooth decay is also important in terms of plaque and sugars in the plaque being broken down by bacteria, and those bacteria then release acids onto the teeth and break down the enamel, right? Once you get inside of the tooth and you get touching those blood vessels, those nerves, that's when you start getting pain. Now let's look at the chemical digestion of food. But first of all, the mechanical digestion, let's look at that. Because mechanical digestion is important in order to have effective chemical digestion. So mechanical digestion is just the teeth grinding and having the food be broken down into smaller pieces so that when the enzymes are acting, they have smaller pieces of food to break down. By having this large surface area to volume ratio, it speeds up how fast those enzymes are able to act on the food. So what is an enzyme? It is a catalyst. It's a biological catalyst, and it speeds up chemical reactions occurring in living organisms. Something that you need to be aware of as well is that there are properties of enzymes, meaning characteristics or traits or things that all enzymes have. One of those is that they are highly specific. They only catalyze one type of reaction. They have an optimum temperature, an optimum pH at which they work best. They are denatured if they are too high in terms of in an environment that is too hot, as well as if they are in a pH that is too acidic or too alkaline, they also would denature. They are also helped when you take certain vitamins and minerals and inhibited by certain poisons like arsenic and cyanide. Now, when we look at examples of enzymes, we see here starch is converted to maltose by amylase, and one that I also want you to pay attention to that is not in the digestive system, but is in all of our living cells, any cell that carries out metabolic reactions, especially highly active cells like liver cells. These have catalase, and this is an enzyme that breaks down hydrogen peroxide. Now, hydrogen peroxide is a natural byproduct of chemical reactions or your general metabolism in your body. And so your body wants to get rid of it because it's a waste product and it could become toxic. So catalase breaks that down to form water and oxygen. How would you see the oxygen? By bubbles forming in the reaction. It's a gas, right? Now, in terms of the enzymes that we have three main categories. We have carbohydrases, which break down carbohydrates. We have proteases, which break down proteins, and we have lipases, which break down lipids or fats. Something about the digestive system I want you to pay attention to is that it really is highly connected. Its structure is connected to its function, right? So, for example, the epiglottis is a muscular tube. It has muscles that contract and relax in order to move the food down into the stomach. The stomach is also muscular to allow for the churning of the food, mixing of the food with the gastric juice. Then you have the pancreas, which produces pancreatic juice, the liver, which makes bile and stores bile, and then once you get into the small intestine area right here, and the first part is called the duodenum, you start seeing lots of villi. Now, these villi we'll get into, but basically the small intestines have a very large surface by having these villi present, right? Especially in the ileum area right here. And why you want a large surface area? To allow faster diffusion, faster absorption, similar like how when you had the smaller pieces of food, you want faster diffusion of the enzymes going in, you want faster diffusion of the products of digestion going in here. Now, when we look at the enzymes that are present, we see the mouth has mostly salivary amylase, or not mostly, only salivary amylase, which breaks down starch to maltose. Then once you hit the stomach, the one you need to focus on is pepsin, which breaks down protein to peptides. In the small intestines, mostly looking at the first part, where the pancreas, remember here, the pancreas led right into this section, the duodenum. You have pancreatic juice, which contains pancreatic amylase, trypsin, and pancreatic lipase. And these break down pancreatic amylase, just like in the mouth, starch to maltose. Trypsin is like pepsin, but in the duodenum. That one breaks down again proteins again. Fresh proteins that did not get broken down in the stomach get broken down here, back to peptides or polypeptides you might see. And then pancreatic lipase, for the first time, fats are broken down in the duodenum. So they weren't broken down anywhere else before the small intestine. Then once you reach further and further down into the small intestine, you start to really have this intestinal juice that is present, just produced by the intestine itself, and this breaks down further the maltose, any sucrose that was there, lactose, and the peptides finally get broken down to amino acids. And these are the enzymes that do those things. Now, once you get to absorption and you have all of the end products available, you can start thinking about, okay, how am I going to absorb these nutrients? Where are they going to go? So I mentioned the villi. So the villi here is the structure of it. And as you can see, just imagine the whole tube lined with these protrusions or these finger-like structures coming out, microscopic of course, right? Now, let's see, it's one cell thick, again, allowing for quick diffusion of the absorbed products. The absorbed products can either go into the absorbed products can either go into the bloodstream directly or into this middle piece. Now, this middle piece is called the lacteal. There is it, right here. And this one is responsible for absorbing all the fatty substances and vitamins that absorb in fat. So fatty acids and glycerol, as well as the vitamin A, vitamin D. But into the bloodstream, you have the water soluble and water soluble vitamins and water, as well as water soluble nutrients. So you have the amino acids, the glucose, um, as well as vitamin C, vitamin B, and water going to the bloodstream. Now, once that is done, you have the products of digestion actually go first to the liver, and the liver is what really determines what happens to those products. So if the liver realizes that, okay, and I think about this like a mother basically monitoring a child eating, seeing, okay, does my child need more glucose right now, need more fats right now, need more protein right now? Does it have enough? So if too much glucose is absorbed, what happens is that the liver will actually try to store some of that glucose as glycogen, or it would send some of the extra glucose to be stored as fat in adipose tissue. If it has, if your diet contained too much protein and therefore too many amino acids were absorbed, it would break them down and convert them into urea, right? And we remember that this is going to be excreted. So really, eating too much protein is not a good thing. You're just going to excrete it. Your liver is going to take care of that. And when it comes to, for example, once your glucose is absorbed and the liver allowed it to pass on, and you didn't have too much glucose, then is when the pancreas will now come into play. Remember this other organ right here that it has two main roles. So it's involved in digestion with the pancreatic juice, but it also is responsible for the balance of glucose in your body, right? So if you absorb a lot of glucose, it goes into the bloodstream, the liver lets it pass through. If you have insulin being produced, you need to have insulin being produced for your cells to then use that glucose, and that insulin is made by the pancreas. So I'm going to stop there. And that whole process of using is called assimilation, using the products of digestion. And then lastly, the difference between digestion and excretion. So digestion is that end just undigested material that didn't get absorbed, a lot of plant-based materials, a lot of fiber that just goes and continues along the large intestine as water is continuously absorbed out of it back into the blood, and that is digestion. So let's look at some questions. So when this question now is looking at the tooth, right? It asks about the types of teeth and the function. So we know that this is a molar and that it is responsible for crushing and grinding food. And then for B, we know that this is an incisor. You have four of them. The canine is the one that is pointed, and we know that these are used for biting off pieces of food. And if I go here to do this one, just looking at tooth decay, we mentioned that bacteria acts with the acid, right? Sorry, not acid, sugar. To form acid, and then those acids break down the teeth and form cavities. Now let's look at this diagram of the human alimentary canal. And this alimentary canal just means a tube going from the back of the mouth all the way down to the anus. So name the structures labeled A and B in figure 4. So A is the, what is it? The liver, right? We see the gallbladder right here as well. And the function of A, we said the function of A was to make bile. as well as help with assimilation of the products of digestion in terms of storing extra glucose or breaking down extra amino acids to form urea. And then with B, the esophagus, we know that that helps with passing the food from the back of the throat or the mouth to the stomach. And the process, peristalsis, is what helps that muscular contraction that pushes the food down. As one contracts one piece. So for example, as the top contracts to push it down, this part is relaxed and it just keeps going. Now let's look at this. They said enzymes are biological compounds are catalysts which speed up the rate of chemical reactions. So this question asks to give the names of two enzymes associated with digestion in humans. So any two enzymes and a function. So you can give any of the carbohydrates, such as amylase, and we know they said that breaks down starch to maltose. You can give trypsin, which is an example of the protease, or you can give pepsin. Please only write one. I'm just showing you more than one now. And we said that both of those break down protein to peptides or polypeptides. Now, next question. The breakdown of food takes place in the mouth, stomach, and small intestine. Name the type of digestion for which the teeth in the mouth are responsible. We mentioned that it was mechanical digestion, or another word for that is physical. Then, humans have 4 types of teeth. State which type of tooth is mainly responsible for the digestion named above. So if they're really talking about breaking them down into smaller pieces, your incisors and your canines don't really do that, right? They just tear it off. So the one you're going to look at, mainly, I would go with the molars at the very back of the mouth. Now next we have some graphs. It says both temperature and pH affect the manner in which enzymes function. Figures 2 and 3 are graphs showing the rate of reaction of amylase over a range of temperatures and pH. So, as I suggested, whenever you have a graph or diagram, please observe it first, analyze it. This says rate of reaction on the Y. On the X, it says temperature for one, and it has a bell curve going up, and similar curve on the other one with rate of reaction on the Y and pH on the X. We see that it has a peak for temperature at around 37 and then a peak for pH around maybe 7.5 or so. And next question. Well, the first question for it, at approximately which temperature and pH does amylase function best? So both of them are looking at amylase. So it wants to know basically this. So around 37 degrees Celsius. And for pH, around 7.5 degrees, 7.5, right? Next question, suggest what effect temperatures greater than 55 degrees would have on enzymes. So if you go greater than 55 degrees here, this is 50, the enzyme is going to denature, right? It's going to break down, and it's no longer going to have that specific lock and key shape. Remember, we said that enzymes are highly specific, so once that shape changes, it can no longer break down what it was designed to break down. Next question says, the consumption of excessive amounts of carbohydrates and fats may contribute to the development of diabetes mellitus. Explain the role of the pancreas in the development of the disease. Now, remember I mentioned with the pancreas, it produces insulin, right? And so this insulin is needed to let glucose into the cells and out of the bloodstream. But if the pancreas stops producing as much insulin as it used to, and sometimes this happens as persons are overweight, as well as their diet, as this is saying here, is high in carbohydrates, and they're not exercising, then they can stop producing or produce much less insulin. The other option that can happen is that they may produce enough insulin, especially in the beginning, but then the insulin is no longer effective. And so it's as if they don't have any, right? So that is how that disease, high blood sugar, can result, leaving the sugar in the blood, not going into the cells. The next question says, suggest two methods other than the use of medication which could be used to manage it. So basically just reversing what was done up above here. Take out the carbohydrates, take out the fats, increase the exercise. This next question, a pancreas becomes diseased and stops producing pancreatic juice. So this is from another question, but I put it here because it's really, again, just showing you how important the pancreas is in producing these two types of substances. So what's one food group whose digestion would be affected if it stops producing pancreatic juice? Well, you just have to think about what the three things were in the pancreatic juice. Remember we said trypsin, which breaks down protein. We also said pancreatic amylase, which breaks down starch. And then we also said lipase, which breaks down fats. So any of these fats and oils would be food group, starches, carbohydrates, as well as food from animals, right? Any of those could be affected. State one effect that a deficiency of the food group would have on the human body. So depending on what you chose, you would have different deficiencies, whether it's protein, energy, malnutrition, when you have a lack of protein and carbohydrates in your diet. Any of those, you can rate for that. Then next question. Figure 5 shows an experiment used to demonstrate the digestion of starch by amylase. Again, observe your diagram. It's showing a water bath at 37 degrees. It has 3 test tubes. Test tube 1 has in starch plus amylase. Test tube 2 has in starch plus boiled amylase. And test tube 3 has in starch plus just water. Now, what do you think going to happen here? Which of them is going to have amylase, sorry, not amylase, starch breaking down? And it's at 37 degrees. It's constant. That's about body temperature, right? And it's an experiment to demonstrate the digestion of starch. So in which of the 3 test tubes would the starch be digested? Well, definitely not in the boiled. We just spoke about denaturing, so no, it's no longer going to fit. Definitely test tube 1. Explain why starch will not be digested in the other 2. Well, if it's denatured, and how much points with this? 4. So I will talk about the fact that if it's denatured, it will not be able to fit to the starch molecule anymore, therefore it will not be able to break it down into maltose, right? That's 2 points there. And then in terms of the water, there's no enzyme to actually break down or speed up the breakdown of that starch. Then, we're soon done. This is the last few. So Vera does not chew her food properly before swallowing. Explain why Vera's way of briefly chewing her food is bad for her digestion. So this is just, again, coming back to the importance of mechanical digestion and increasing or having a large surface area to volume ratio so that you can have more slash quicker diffusion of the enzyme into the smaller pieces of food to break it down chemically. In which region of the digestive system would the final stage of digestion of starches occur? So remember we said that maltose was made by the amylase, sorry, was the end product of amylase enzyme, right? And then maltose has to further be broken down into what am I saying? Yeah, into glucose, right? So by maltase. And this is going to take place in the small intestines through the intestinal juice, as we saw earlier. Now, last question. I think I am going to end at this one. The villus. Now, we saw this in the book. It gives you a nice little diagram here: capillary, arterial, venule. Remember, arterial and venule are just small versions of a vein and an artery. It says, using the diagram, explain how villi are well adapted for absorption. So, how are they well adapted? Structure. How does the structure help with it doing its role? We said it's then only one bare cell here. Quick diffusion. Bam, that's one. Then it contains a lot of capillaries. Why you want to have a lot of capillaries? So that it can be transported into the bloodstream and go throughout the body quickly, right? So that the cells can use the end products of digestion. That's another set. It also has a special lacteal, which is actually part of the lymphatic system, and it takes away the fats, the fatty acids and glycerol, which are the end products, right? Vitamin A, vitamin D. And so this also makes it well adapted for absorption. And last but not least, because of this great folding shape, it increases big time the surface area to volume ratio of the small intestines, giving you a larger amount of space for absorption. State one: process by which the nutrients are absorbed into the bloodstream. Well, one that you always have to remember: diffusion, moving from high to low concentration. Alright, so I hope this was helpful, and let me know if you want me to do any others for another topic.